What's up, gang? It's Ashlyn Wilder, and I'm here today with
Brooke Fitzpatrick.
and
Katia Shushkina.
I just hear the elevator. Today I'm going to be asking the age-old question, how does DNA replicate? The structure of the DNA molecule.
Of course. So first, let's start with the sugar phosphate backbone. The sugar phosphate backbone is the structural bonded framework of DNA and RNA. It is made of alternating sugars and phosphate groups. Here you can see the black pentagons of the DNA sugar and the white tubes of phosphate. Nucleotides are next. They are the functional units or building blocks of nucleic acids. Each contain 5 carbon sugar, a phosphate group, and a nitrogen base. Nucleotides are linked together to form nucleic acids. In our structure, it consists of the grouping of the phosphate, sugar, and the nucleic base. So over here, you can see the black pentagon, white tube, and then our base. The 4 DNA nucleotide bases are adenine, guanine, cytosine, and thymine. In DNA, adenine must be linked with thymine, cytosine must be linked with guanine. In RNA, adenine must be linked with uracil, and cytosine must be linked with guanine. Hydrogen bonds are weak, non-covalent attractions between the nucleotide pairs that hold the DNA strand together. Here you can see the clear rods that hold the pairs together.
Can you show me and tell me how DNA replication begins?
Okay, so how it begins is a portion of the DNA unwinds and unzips like a zipper. Also, we can determine the leading strand and the lagging strand by looking at the phosphate and which direction the DNA reads. Over here we can see that this is the DNA, or the leading strand, because our phosphate's over here and it's reading. DNA hydrogen bonds must be broken between the nucleic bases in order for DNA replication to begin. This unzipping is carried out by an enzyme called helicase.
So what name do we give the shape of the DNA when it's unzipped and ready to replicate?
The shape of the DNA is described as the replication fork, as it separates and begins to replicate because it looks like a fork.
So the leading strand replicates by starting with a primer at the bottom, and it goes from 5 to 3, and it builds upwards. And then once it hits that halfway point, then we start.
Can you tell me how the leading strand replicates?
So DNA polymerase synthesizes a new strand moving towards the replication fork.
Show and tell us how the lagging strand replicates.
So the lagging strand replicates in short segments called Okazaki fragments, moving away from the replication fork. Alright, so then for the lagging strand, it begins in the middle and it builds its way down.
Because it's going from 5 prime to 3 prime.
Just like that.
And then we start at the top again.
There we go, with the primer being at the top. With the primer being at the top and building its way down again.
It starts going up again.
Yeah. And then we go back into the leading strand. And that begins to build upward again. Perfect. Connects, and then
We have to leave this gap right here between these two because this will be filled in later at the very end. Additionally, ours doesn't have uracil in our RNA because we didn't have a green adenine as like the base, so we couldn't add a uracil as the RNA base. So yeah, this is how it looks so far.
What's the name of the key enzyme that adds the new DNA nucleotides on the lagging strand?
it is DNA polymerase.
What are the final 2 steps of DNA replication?
Alright, the final steps is that we need to get these RNA primers and we need to replace them with the DNA nucleotides. So we would get these bases and we would replace them with the nucleotide version, and we would get rid of these RNA sugars and also replace them. So we just replace them all right now. And at the very final step, once everything is replaced, then we can finally stitch together this gap over here with the protein ligase. So now we can finally stitch together this gap And now we have our 2 duplicates of the DNA.
Wow.
Wow, I just learned so much. My brain is so much better, or bigger. That was signing off.
Bye-bye.